The discourse surrounding the safety of COVID-19 mRNA vaccines has reached a critical juncture, particularly with respect to their potential harm to the central nervous system, or CNS. The proliferation of CNS-related adverse events is a significant concern that's been linked to mRNA vaccines. A study titled COVID-19 mRNA Vaccination: Implications for the Central Nervous System was uploaded to Zenodo around September 18, 2025. This study analyzed reports from the Vaccine Adverse Event Reporting System, or VAERS, from January 1990 to November 2024. The findings are striking. The study identified 63 serious CNS-related safety signals, including rare neurodegenerative conditions, infections, and inflammatory syndromes, with incidence rates far exceeding those associated with flu shots. The key statistics include neurodegenerative and demyelinating conditions, such as Creutzfeldt-Jakob disease, an otherwise rare disease, and it was reported 847 times more frequently after mRNA vaccines than after flu shots. Myelitis of all types was 31 times more likely, with specific types like transverse myelitis and non-infectious myelitis showing dramatic increases of 25 times and 132 times more, respectively. CNS infections, like all types of meningitis, was 34 times more likely, with aseptic meningitis at 53 times higher and bacterial meningitis 36 times higher. Autoimmune encephalitis and limbic encephalitis showed increases of 79 and 146 times, respectively. Herpes zoster meningitis was over 1,200 times more likely, with other herpetic CNS conditions like meningoencephalitis at 136 times and neurological disease at 680 times more also spiking. Then there were conditions such as brain abscess, 120 times more, and extradural abscess, 169 times more, were significantly elevated. These figures suggest a pattern of neurological disruption that has alarmed researchers and the public alike, especially given that many of these conditions are rare, and their association with vaccination was previously undocumented at this scale. There's a point that I want to make here. The condition that is most telling to me is the aseptic meningitis, which is inflammation of the membranes covering the brain and spinal cord that is not caused by a bacterial infection. Basically, it's one of those things that we don't have a clue what causes it, so basically they don't know, which is ridiculous, and it tells me two things. First, that there clearly is an issue with these vaccines passing the blood-brain barrier, and secondly, that it appears that a component of the vaccine could very well make it hypersensitive to anything that could cause inflammation, potentially even stress. It somehow appears to impair the integrity of the blood-brain barrier, which scares the heck out of me. The study itself proposed a mechanism that is rooted in the action of lipid nanoparticles, LNPs, which I have discussed in detail in a prior video that I did over a year ago, and now the proof is coming out. I will be linking that video at the end, but the delivery vehicles in mRNA vaccines are designed to increase distribution, and they do. Let me make an association. It's a goal in chemotherapy to limit distribution only to malignant cells so as to minimize collateral damage to healthy cells. Well, it's the same thing here. The mRNA vaccine has a lipophilic vehicle that carries the drug into fat stores, creating a non-specific distribution into areas that the vaccine doesn't need to be. So for me, a major problem is that they are not specific and infiltrate our brains and other organs, as you will see, which is not where immunity occurs. So now the research indicates that lipid nanoparticles can cross the blood-brain barrier, which is the protective layer that regulates the passage of substances into the brain and spinal cord. Once inside, the spike protein production appears to trigger a vascular or blood vessel inflammation, and that's how it disrupts the blood-brain barrier's integrity. Studies have supported this theory, but it's a very basic concept of solubility that any legitimate researcher should understand, and to be honest, it almost sounds like the lipid-soluble nanoparticles were designed to go places that they shouldn't. The extensive distribution into the nervous system and vascular inflammation is totally predictable. It wasn't a mistake. This disruption can be assumed to make the brain more vulnerable to infections and inflammation because the compromised BBB could allow pathogens or immune responses to enter the CNS through the damaged protective membrane. Prion-like behavior is also expected, which means that the spike protein exhibits properties similar to prions, which are misfolded proteins that induce further misfolding. We saw this in mad cow disease, and this could explain the elevated incidence of prion diseases and the formation of amyloid-rich clots that have been seen in some autopsies. A related study by Thorpe et al. Linked the mRNA vaccines to increased mental illness also, including 86 neuropsychiatric disorders, including dementia, psychosis, and suicidal thoughts, potentially tied to this CNS inflammation. The infiltration of the brain by any substance is a reasonable concern. These mRNA vaccine risks were often dismissed by mainstream bodies like the CDC and FDA as lacking sufficient evidence, but the risk is very real when you understand how the body works. Here's the scary thing to me. The vaccine manufacturers have deemed this accumulation consistent with the mRNA vaccine design. And what concerns me most is that they didn't consider these levels indicative of a high risk for infertility or other harms. The FDA and EMA maintain that there's just no evidence from regulatory reviews or from post-marketing surveillance systems, but I say they are either lying or deliberately looking in the wrong places. Neurological symptoms aren't the only concern either. It's now widely admitted that the vaccines are squarely related to an increase in myocarditis. In myocarditis, the myocardium, which is the heart's muscular wall responsible for pumping blood, is inflamed. Now picture this, guys. The entire purpose of the heart is to pump blood, and the muscle responsible is inflamed. So far, that's all we know. We don't have the studies to determine if it also necroses or dies. Now, the myocardium is primarily composed of cardiac muscle tissue, not fat. But guess what? The heart is also protected by lipids. The epicardium and pericardium both protect the heart, just like the blood-brain barrier protects the brain. The serous pericardium is made of mesothelial cells with lipid-rich cell membranes called phospholipid bilayers, and the epicardium also contains a thin layer of adipose or fatty tissue, particularly around coronary arteries. But the plot thickens. Many protective barriers in the body are membranes which contain or are composed of lipids. So if someone really wanted to screw up as many human organs as possible, bypassing lipid barriers is actually quite an ingenious way to do it. Because listen to this, guys. Several critical organs are protected by lipid-based barriers. Notably, the brain, heart, and all nerve cells, which we've discussed. We also mentioned accumulation documented in the liver, which is another organ protected by lipid structures, along with the lungs, kidneys, the eyes, and the spinal cord. Let me tell you why I am concerned about additional delayed adverse effects. Once something gets through the barriers of the heart and brain, there's almost immediately perceivable events. It's different with the kidneys, liver, and lungs. These organs are more of a filter. They might remove oxygen or metabolites and toxins, but as they begin to break down, these organs usually become less effective in a gradual manner. So when we see the vaccine metabolites accumulating in these organs, we will likely notice a more gradual reduction in the performance of these organs. The same with the eyes. Contamination of the eyes could result in a gradual loss of vision. When irritants bypass the pleural barrier into the lungs, they trigger inflammation, airway constriction, or alveolar damage, potentially causing symptoms like coughing, wheezing, or reduced oxygen exchange. Chronic exposure may lead to infections, fibrosis, or conditions like COPD. And what a shock! I did some research on side effects involving the lungs and eyes with the mRNA, and of no surprise, blurred vision, optic neuritis, uveitis, and retinal vascular occlusions have occurred. Also, breathing difficulties have been identified, such as shortness of breath from interstitial lung disease, pneumonitis, pulmonary embolism, or acute respiratory distress syndrome. So these vaccines have shown adverse events in all critical organs that are protected by lipid membranes. The only organs that are not protected by a lipid barrier is the spleen, pancreas, thyroid, and bone marrow. Notably, there have been very rare side effects in these organs from the mRNA vaccines, less than 1 in 1 million doses, which is statistically insignificant and a whole lot lower than the lipid-protected organs. So I'd say our experts need to get on the ball with this mRNA technology and begin teaching people to heal with food, stress reduction, and movement. Also, there are allegations that biodistribution data submitted by Pfizer to the U.S. Food and Drug Administration for its mRNA-based COVID-19 vaccine was edited or manipulated. What a shock this is, huh? Biodistribution studies document how a vaccine's components, like the lipid nanoparticles, spread throughout the body after being injected. This data is crucial for assessing safety. It also examines potential adverse or off-target actions, such as the accumulation of a substance in organs like the ovaries, liver, or spleen. This is standard bioavailability data required for the approval of all drugs, and to fudge it is unconscionable. Personally, I think that if any such falsifying of data is discovered, the manufacturer should not only be fined, but they should lose their license to manufacture drugs if it is found to be linked to upper levels of management. Once a full-scale investigation is launched, they have found the root and removed them. Then they can reapply. The claims stem from a non-clinical study conducted by Pfizer's Japanese partner Keyclide in 2020. This study, known as Study 185350, used a surrogate marker. Guess what that surrogate marker was? It's called Luciferase mRNA. In 2021, it was adamantly denied that the vaccines were manufactured with luciferase, which is bioluminescent marker that lights up and tracks how the vaccine is distributed throughout the body. Besides substituting luciferase mRNA for the actual spike protein in the vaccine, the study was non-GLP, or good laboratory practice compliant, meaning it lacked full standardization for regulatory toxicity assessments, yet it was used for safety data. Also, the study involved very limited sample sizes, like only 3 mice per group, which did not compare to the millions who the vaccine was given to. There's been a lot put out about luciferase because its name originates from the term Lucifer. Interesting, isn't it? There are a lot of claims out there about luciferase being part of the mRNA vaccines. Controlled media outlet USA Today reported that luciferase enzyme is not in the vaccine, and it has nothing to do with Satan. I haven't found evidence yet that the enzyme is included in the vaccine, so we can't go there, but it does appear that they lied about using it in the development, so how do we know what to believe? So let me share what we can find in the data. Allegedly, the luciferase enzyme does not appear in any known approved medicines. It's mainly used in research and diagnostics to track gene activity or test drugs in labs, not as a therapeutic in human treatments. They tell us that there are no FDA-approved luciferase-based drugs for human clinical use because introducing foreign genes or enzymes carries risks like immune responses or unintended effects. Yet we are seeing an unreasonable and ignored occurrence of such inflammations in the brain and the fact remains that all vaccines are injected to stimulate immune responses, which cause inflammation. I find this quite interesting. There are many purported reasons why luciferase is not a good diagnostic tool in humans, though it is used in animals. So we always need to look behind the official narrative. In preclinical research, which is science done in labs before testing on humans, luciferase is often used as a glowing tracker to study how genes or treatments work. Scientists put the luciferase gene into cells or animals like mice, using tools like plasmids or small DNA loops or viruses that act like delivery trucks. When the luciferase gene turns on, it makes the luciferase protein, which glows when a special chemical is added. So why is this substance named after Lucifer, the fallen angel? It was named by a French pharmacologist allegedly from the Latin word Lucifer, which means light bearer, and it comes from those beautiful little lightning bugs that kids used to pull off and put on their finger. Okay, so that's the premise of why it glows and using it in bioluminescence, but why didn't he name it lanternase or lucinase? It is widely known that Lucifer refers to Satan, the fallen angel, so why in the world would he pick such a name? While I was unable to find a direct connection by its inventor, Raphael Dubois, I did learn that there was a French occult revival centered in Paris, which peaked in the 1880s to 1890s. Dubois named the group of bioluminescent substances luciferase around 1885, so the name could clearly be based in the black magic or dark arts. These findings come against a backdrop of evolving vaccine safety debates. In comparison, historical vaccine safety concerns, such as the 1976 swine flu vaccine linked to Guillain-Barré syndrome, led to swift investigations and program adjustments, but they just kept pumping out and mandating the vaccines in spite of apparent adverse effects dating as far back as the studies. The current situation has been further complicated because the scale of mRNA vaccine deployment was billions of doses globally. The potential liability can be unprecedented if determinations are made that the risks were suppressed or the vaccine lacked sufficient safety studies. But remember, they are banking on the emergency nature of the COVID-19 pandemic. But personally, I believe that it is easy to prove that the communicability and course of the disease was grossly inflated to justify the expedited emergency vaccine distribution. Personally, I'm concerned about why President Trump continues to tout that vaccine as a success. I totally get it that he was lied to by the experts, Fauci and Birx, and likely many other deep state-owned scientists. I'm sure they showed him bad medicine, and as any good leader, he delegated responsibility to people more educated in medicine and immunology. But apparently, that can work two ways when you have cabal-owned experts that have made commitments to depopulate the earth to what they believe is a manageable size. Such initiates requires compassion and a sense of right, which I don't believe those who have been in control have. Some might say that POTUS's ego is too big to admit a mistake. That's a possibility, but I think there's much more to his madness, so to speak. RFK Jr. has restricted mRNA vaccines through several actions in 2025. He canceled nearly $500 million in federal funding for 22 different mRNA vaccine development projects that target respiratory viruses like COVID-19 and flu. He also halted all new mRNA-based research and investments by BARDA, and he is influencing FDA approvals to limit updated COVID-19 mRNA shots primarily to adults 65 plus and high-risk individuals under 65, which eliminates a large portion of Americans. Additionally, his appointees to the Advisory Committee on Immunization Practices have been told to drop broad CDC recommendations for COVID-19 vaccines, framing them as an individual decision rather than a routine endorsement. Of course, critics hate this. All he's doing is saying that we shouldn't push them as routine treatments and Big Pharma is creating fear. These moves, justified by Kennedy's claims of mRNA risks and inefficacy, have drawn widespread condemnation from some scientists for potentially weakening pandemic response capabilities, but personally, as an ex-pharmacist and an ex-drug regulator, I am in 100% support of Kennedy's efforts to stand up to Big Pharma. Add that to President Trump's resounding "don't take Tylenol" admonitions at today's press conference, I'd say that Big Pharma is none too happy with this administration. There's a new sheriff in town, and it's clearly putting the safety of Americans over corporate greed and profit.